I'm George Mattox, and I'm going to be talking to you guys about some fishes from the Amazon, and that are among the smallest vertebrates on Earth, actually. Oops. How do I? Oh, here you go. So just for starts, of course, I study the Styrofasins, the largest freshwater fish group in the world. You might be familiar with the Cypriniforms and the Siluriforms, but I think that most of you are not acquainted to the Classiforms, which is the group that I'm going to be talking today. So Classiforms belongs to the Eutophysin, among Cypriniforms, Siluriforms, and Gymnotiforms, and they all are defined by the presence of the Weberian apparatus. So Classiforms is an order that occurs nowadays only in in the Nootropics, especially in South America, and there are 200 or 300 species from Africa. But here in Nootropics, and especially in South America, they are very, very diverse. They have more or less 2,200 species. And they occupied approximately the same niches that Cypriniforms occupy in other parts of the world. So we don't have Cypriniforms in South America. We have Classiforms instead. So Classiforms include the famous piranhas, the pencil fishes and headstanders and hatchet fishes. Some of you might know them from the trade. And I'm gonna be focusing on the largest subunit of Classiforms, which is the family Carassidae. Carassidae nowadays comprises more than 1,200 species, and they are very colorful, very beautiful fish, and they are very famous in the aquarium trade. So most tetras from the aquarium trade belong to the Carassidae. And narrowing down, I'm going to be talking to you about, I'm going to use my PhD on the subunit Caracini, which is a subfamily of Carassidae, to start my talk today. So as a PhD student back in 2006, my advisor, Monica Toledo Pisa, and I decided to study the phylogenetic relationships of this subunit of the Carassidae, which is the subfamily Caracini. So it's just a regular morphological study. It took me like 3 or 4 years to conduct a full osteological study, and also I included a few myology characters. And after 4 years, I came up with this phylogeny of the group, which already shows that the group was not monophyletic. So there's this subset highlighted in red, in the red box, which actually is the real Caracini, because it holds and includes Carricks, the type genus, not only of the family, but of course, of Classiforms as a whole. But I'm not going to talk about this subset. These are mostly carnivorous or lepidophagous fishes of medium-sized, small to medium-sized. But I'm going to focus on this other subunit that we actually extracted from Caracini in this phylogeny. And this subunit in blue here, or green, it's called the Heterokarasini, okay? And it is comprised basically by very, very small fish, except for Loncogenes, which has, 10 or something between 5 and 10 centimeters. most of the other Heterokarasins are very tiny fish. And of course, there's Priorcarricks, which is the main focus of my talk today. So when I was doing the phylogeny of the Caracini, and I included Priorcarricks. So this is, for instance, the phylogeny, the phylogenetic relationships of the Heterokarasins without Priorcarricks. When you include the Priorcarricks in the landscape, in the stage, you notice that the whole relationships collapses. So this is where Priorcarricks aerial shows up in the tree, doesn't help it a lot. So that actually bothered me a lot. And I started wondering why Priorcarricks messed up with the phylogeny so much, right? So I dug up in the literature, and I saw this quotation mark by Weitzman and Bari, quotation by Weitzman and Bari when they described Priorcarricks. And they say Priorcarricks is an example of the problems inherent in studies of the phylogenetic relationships of miniature pedomorphic species. Plesiomorphic features can be distinguished from pedomorphic characters only when phylogenetic hypothesis and developmental information for characters in in-group and out-group taxa are available. And if you look here, there's the part of my matrix showing that most of the characters for Priorcarricks were actually coded as inapplicable, either because the bone were not there, or because the bone was so different that we couldn't code a proper state of characters. So this might be the reason why Priorcarricks collapsed the whole clade. So I finished my PhD at this point, and I started to dedicate the next few years of my life studying Priorcarricks. So Priorcarricks was described in 1987 by Stan Weitzman and Rich Vary. They described the genus and 2 new species. And they define Priorcarricks based on the larval rayless pectoral fin in adults, as you can see in their original illustration. And that's actually not very common among fishes. As far as I know, Priorcarricks is the only genus in Classiforms that actually has this larval rayless pectoral fin in adults. In addition to this striking feature, Priorcarricks is also defined by numerous conical teeth on the dentary and on the upper jaw, as you can see. And of course, its diminutive size. So this is Priorcarricks on the tip of my finger, and this was shot by Ralph back in London. And there's also, a few heuristic characters supporting the, I won't say monophyly, because at this point, Priorcarricks wasn't included in a formal phylogenetic analysis. But they helped to diagnose Priorcarricks, especially the 5 pelvic fin race, because Classiforms usually have 7 branch pelvic fin race, with few exceptions, and some characids, very small ones, would have 6, but not many species of characids have 5 pelvic fin race. So that's something very interesting. So continuing with the paper by Weitzman and Bari, as I mentioned, Priorcarricks was described with 2 species. So Priorcarricks aerial from the upper Rio Negro. So this is, you might not be quite acquainted to this. So this is a map of rivers from North and South America, basically the Amazon basin. And this is the Orinoco basin. And they are connected, the Orinoco and the Amazon basin, through the Caziqueri channel that connects the Orinoco and the Negro system, which is an affluent of the Amazon basin. So Priorcarricks aerial, the first species, is described from this region between the Negro system and the Orinoco system. And the second species, Priorcarricks pygmeus, was described from the borders between Colombia, Peru, and Brazil in a city called Leticia, which is actually in the Colombian side. Okay, so that's it. Basically, we had until very recently only these two species. And I'm going to start talking about how we discovered a third species, also from the Negro Basin. So we wanted to answer that question and try to get fresh specimens of Priorcarricks to study their osteology and the myology in detail, because one of the things that made me code so many inapplicable characters is because museum material of Priorcarricks was very, very in poor shape. You can imagine they are very tiny and they are very fragile. So basically scales are lost, fin rays are broken, and of course they don't clear and stain very well because they've been sitting in museum jars forever and they were not prepared or fixed properly. So I got in touch with Rolf Britz, who welcomed me in his lab, and we started developing this great collaboration. So back in 2011, we organized an expedition to the Negro to collect more Priorcarricks for our study, because we wanted fresh collected specimens in order to do our osteological study. So of course we rented this boat. We spent maybe around 10 days, between 7 or 10 days. There were two expeditions to the same place. Ralph joined for the second one. And of course, we collected several species. In this occasion, we collected the blue belly night wanderer that Professor mentioned in the beginning of my talk. But that's going to be a theme of another lecture. So we actually caught Priorcarricks, and we were successful in sampling several specimens of Priorcarricks. But when we went back to the lab, it was clear that we had two distinct forms of Priorcarricks. Okay, the first one was clearly distinct from Priorcarricks aerial, because we went there in search of Priorcarricks aerial, and we caught something else very different, very distinct from Priorcarricks aerial, and more restricted in their distribution, and but with several interesting characters. For instance, their pelvic fin has 6 branched fin rays instead of 5. There are always some meristics that were, slightly different. And also another striking feature was in the vibarian apparatus, because I didn't say that before, but Priorcarricks is the only crassiform that lacks the claustrum. That's the first ossicle from in the vibarian apparatus. But this new form had the claustrum, and as I said, had 6 pelvic fin rays, branch pelvic fin rays. So we were certain that we had this new species, and we described it as Priorcarricks nanus back in 2014. So other osteological features also helped to identify and separate Priorcarricks nanus from the previously known Priorcarricks aerial. I've already talked about the lack of claustrum in Priorcarricks aerial and pygmeus, and you can actually see in Priorcarricks nanus a tiny claustrum here. Another striking difference between Priorcarricks aerial and Priorcarricks nanus is that Priorcarricks aerial has, very few skeletal ossified elements in the pectoral girdle. Basically, only the endoskeletal is present, but you still have a scapulocoracoid cartilage, doesn't develop into scapula and coracoid. But you see here in this illustration from Weitzman body that you don't see a postclythrum here, whereas in Priorcarricks nanus you clearly have two postclythrum. So that's another striking difference between these two species. So we described it back in 2014. As you see, and I mentioned before, it is very, very restricted, so it's not very easy to find Priorcarricks nanus. And that's it. That's the third species described in the genus. Now I have to take a detour, a small detour, to show what we wanted to do with this Priorcarricks. So I'm not sure if you're familiar with this technique, or actually it's a kind of analysis of osteological data, where you can actually look at the ossification sequence of a given fish. Or actually, this is done for other vertebrates as well. So this is a graph developed mainly by Paul B. And here in this axis, you have the length of the fish. And in this column, you have, the many different bones that develop. And you can actually see, and they did this for the zebrafish, the model in development. They actually came up with a sequence of ossification for the zebrafish. So these bones in the upper left corner are the first ones to develop. And as you go down the graph, you see the last bones to develop in the osteology, in the skeleton. Basically, this is the cranium, and these are the paired fins, and sorry, the paired fins are this in this study. These are the unpaired fins and axial skeleton, showing the same pattern, right? So you organize the bones according to their size in development. So we thought, very cool, right? Moreover, Ralph Britz and Kevin Conway used the sequence of ossification of Daniel Rario to code the osteological losses of the recently described Pedocypris, the smallest vertebrate, and it's from the Southeast Asia. So this is their cranium, their skull, showing, of course, lots of cartilaginous parts that won't develop. So when you map these losses in the skeleton onto the generalized ossification sequence of a zebrafish, and they did that by painting the bone losses in gray, you see that pretty much the bones that are missing in Pedocypris are towards the end of the ontogeny. Of course, you have a few exceptions here, but most bones that Pedocypris lack start developing late in ontogeny in the cypriniform. So I fell absolutely in love with this paper, and I wanted to do that for Classiform. So I did my postdoc with both Monica and Ralph. As I said before, Ralph was very kind to receive me in his lab back in London. This was 2010, almost 11 years now. And so we picked Salminus, which is a big caracid, very common in fish farms here in Brazil, to settle that as a model of our sequence of ossification. So the first part of this postdoc project was determining the sequence of ossification of Salminus brasiliensis. So that's it. We were quite successful to do that. Here are images showing some of the anatomical complexes that we actually studied. And then after that, after the model of sequence of ossification for a Classiforms, for a Classiform was done, we then went back to Priorcarricks and coded all the bones lacking in Priorcarricks onto the developmental sequence of ossification that we had just produced. And of course, Priorcarricks is not as truncated as Pediocyprus from my previous slides, but the few bones that are lacking in Priorcarricks are also from the late ontogeny of a Classiform. So we had in Priorcarricks pretty much the same model of developmental truncation as we saw in Pediocyprus. So these are just the list of missing bones in Priorcarricks and also a small list of skeletal simplifications. We only work with missing bones for this analysis. And then, of course, we wanted to know the exact place where Priorcarricks would fall, because once we knew that the bones missing in Priorcarricks were not plesiomorphic, but rather apomorphic conditions due to developmental truncation, we needed to reevaluate the phylogenetic hypothesis. So cladograms A, B, and C show actually Priorcarricks in a basal position were coded as with all the missing bones as plesiomorphic. Okay, a condition, a plesiomorphic condition. But we also coded the missing bones of Priorcarricks as derived conditions based on the development of Salminus. And for our surprise, Priorcarricks was no longer a rebel destroying the clades, but nested very nicely with Nathocarricks, which is this also small caracid here. And the whole Heterokarasins were again well organized. So that was actually very good. But there's more. Remember, I mentioned before that when we did the field trips to the Rio Negro, we caught clearly two forms of Priorcarricks. One of them was Priorcarricks nanus that I talked about previously, and the other one we tentatively identified as Priorcarricks aerial. So this is another great photograph by Ralph Britz. Note that Priorcarricks is basically translucent in life. You can actually see the swim bladder, the heart, the liver, and all the internal organs. And we actually saw that Priorcarricks aerial from Rio Negro had greatly overlapped the meristic counts of Priorcarricks aerial from the type locality, right? These are data from the original description. So basically, both forms have the pelvic fin rays, 5 pelvic fin rays, the number of dentary teeth also overlap, the number of gill rakers also overlap. So basically, we were dealing with Priorcarricks aerial. But when we clear and stain them, we were surprised. So once again, I highlight the pectoral, the rayless pectoral, larval pectoral fin of Priorcarricks aerial from the type locality in Venezuela. And remember, I mentioned there are no postclythra. Priorcarricks aerial from the Rio Negro has a single postclythra here, postclythrum. Remember that Priorcarricks nanos has this postcliatum and then another postcliatum. So there's a variation there. Another source of variation lies within the infraorbital series. So from the original description, Weitzman and Vadi mentioned that infraorbital series are incomplete, with antorbital bone only element ossified. But that's not what we saw in Priorcarricks aerial from the Rio Negro. In addition to the antorbitals, we also have 3 infraorbital bones. Classiforms usually have 6 infraorbital bones. So that's actually a reduction from the model, right? From the rule of classiforms. But still, they have 3 infraorbital bones. And another thing that also caught our attention was that males of crassids usually have pelvic fin, anal fin hooks, bony hooks, in the mature males. So here you can actually see that Priorcarricks ariel also have these tiny bony hooks on the fin rays of the anal fin. And but some carassids also have these hooks on the pelvic fin. Okay, in the description of Priorcarricks ariel, Weitzman and Vadi state that anal hooks are present in a few specimens which were mature males, but that no hooks were present on the pelvic or other fins. When we studied the Priorcarricks ariel from Rio Negro, we also saw the hooks on the anal fin, but the pelvic fin also had hooks. So back in 2017, we came up with this table, a tentative table, right? To try and figure out this variation of characters. So we had Priorcarricks ariel from the type locality, Priorcarricks pygmeus, Priorcarricks nanos that we had this recently described, and we had Priorcarricks ariel from the Rio Negro. And as I mentioned previously, most of the meristic characters of this form of the Rio Negro overlap with Priorcarricks ariel, but some osteological differences caught our attention and are different, right? So in addition to this little story, of course, we increased sampling efforts, both in areas relatively known and also in remote localities in the Brazilian Amazon. And with that increase in sampling efforts, a new scenario has been reviewed referring to the diversity of Priorcarricks. So basically, we had the 3 species, ariel, pygmeus, and nanos, but some literature started to appear in 2010, 2011, registering the presence of Priorcarricks ariel in other places, such as the lower Rio Negro, an affluent of the Solimões River, and also in the Madeira system, which is, one of the largest tributaries of the Amazon. So that's pretty weird. Also, I organized new field trips to the Amazon, and we actually sampled all these blue spots here. So there's, lots of new records of Priorcarricks. And of course, looking into the collections, we found that Priorcarricks was more widespread than previously thought. Okay, and the reason that this was only evident in recent years is because I have a project focused on these small fishes. Because if you look, if you go to the field just to make an inventory or to generally collect fish, these small miniature fishes easily passed unnoticed, either as baby juvenile fry fish, or, people actually discard them because, they think there's no value. They won't they won't be able to tell the taxon that they're dealing with. So it's common to actually see Priorcarricks identified as, uh, juveniles of other genera of crassids. So you need, in other words, what I'm trying to say is that you need specific efforts focusing on this miniature taxa in order to actually do your study, right? Because trusting collections only will only lead to more questions rather than more answers. So the taxonomic problem is far more complex than originally expected. For instance, the form from the HUMA data, I went there in 2017, and we actually caught the specimens, caught a handful of specimens. And we noticed when we went back to the lab that Priorcarricks had a small adipose fin, this form of the Madeira, right? All the other 3 species of Priorcarricks actually lack the adipose fin. Actually, that's actually another case of truncation in Priorcarricks. But this guy had the adipose fin. 50 plus specimens show this feature. And also, this form has 6 pelvic fin rays, just like Nanos. So and of course, the cherry on the cake was the molecular data, because it clearly shows that this is a different species. So we recently described that species as Priorcarricks vari in honor of the late Richard Vari, one of the descriptors of the genus Priorcarricks. But as you said, as you saw already, it's the thing is more complex. So right next to the Madeira system, we have the Purus system. And in the same field trip, we were, just a couple of hours away from this Purus river. So we decided to get the car and go there and sample the Purus system as well. And we caught this other form of Priorcarricks, very, very translucent, doesn't have much of a color pattern except for the tiny line of chromatophores along the anal fin base. And we cleared and stained that. And of course, we took several measures. And it turns out that this is potentially a new species as well. What differs is that in addition to the antorbital, it has 2 infraorbitals, as you can see here. So it's different from aerial, it's different from nanos that also lack, vari and nanos lack the infraorbitals. And also in the pectoral girdle, it has a huge postclathra here, and another very tiny and very thin postclathra 3 with a different shape, because usually postclathra 3 in Classiforms is just straight. And in this new species of Priorcarricks, it has, this sinuous form. Okay, but that's actually not described yet. Also, and until recently, Priorcarricks was known from the heart of the Amazon jungle, the central portions and the upper portions of the basin. And recently, Priorcarricks started to be sampled in the far east of the Amazon basin, near the Amazon mouth itself. And so this is the rivers draining Amapá state. This is actually the Amapá state. So I went there and I caught Priorcarricks, many specimens. They are actually quite huge for Priorcarricks. Of course, they are miniatures. They don't grow over 26 millimeters, but compared to the other species, they are more robust. And on the other side of the Amazon, so this is this photograph was taken in this upper red dot. But then I had material from collections showing that in this specific spot, there were a form very similar to the Amapá form, which are, A and B here. But there's also another form, very, very rare in collections. Actually, I have only 3 or 4 specimens. And they are very different. They differ in a number of features. And I'm sure it's not a sexual dimorphism because we have males and females of both forms, okay? But this thing here, the highlighted in C, has, a very robust hook fins, pelvic hook fins, anal fin hooks, and it's very bizarre. So because we only knew this from collections, I organized 2 field trips to this point, to this spots. We were not successful in sampling Priorcarricks in the first field trip. And then we tried the second field trip, and I had the great pleasure to have Kevin Conway joining me for this field trip. And this is actually the first footage that Kevin made, and it's available for Priorcarricks. So I wanted to show you how the rayless larval pectoral fin moves, and that's it. So this is pretty much I think it's the only film of Priorcarricks ever. So another step that we did, in addition to the description of this different species that are actually underway, we tried and mapped the developmental truncations of each species on that model that I showed you before. So this is basically the same diagram, but separate in the different anatomical complexes. And this clearly shows that Priorcarricks, no matter which species, has developmental truncations in the different complexes. So first, for instance, in the infraorbital series, Priorcarricks aerial from the Rio Negro has 1, 2, and 3. So this is the red arrow points. The other species either lacks the infraorbital series completely, like Priorcarricks aerial and pygmeus and nanus, and vari. Okay. Another thing is the claustrum that I've already told you about. So Priorcarricks nanus is the only species that has the claustrum. The other ones lack the claustrum, as inferred by this gray bar. And at last, in the pectoral girdle, there's this thing going on with the postclythrum. So Priorcarricks aerial has 1 postclythrum, where Priorcarricks aerial from the Rio Negro, sorry. Priorcarricks aerial, the true one, and pygmeus lack entirely the postclythra, but Priorcarricks nanus and Priorcarricks vari have 2 postclythra. And when I mention that, I must say that this is consistent. We have, several specimens clear and stained, and all of them show this exact same pattern. And of course, I'm not showing here the same graphs for the undescribed species, but they follow the same pattern, which is each species, even though their meristic and morphometrics overlap to some extent, you find osteological differences relating to their developmental truncation. Or in other words, each species is actually defined by a mosaic of different developmental truncations. So that's something quite unusual to my knowledge. And of course, as I mentioned before, the molecules also corroborate the species. And in addition, they not only corroborate the identity of the species so far described. Of course, I have unpublished data supporting the idea that each of these lineages are also new species, but also their pardon, their genetic differences is so it's huge, right? So it shows that either the different lineages of Priorcarricks are separated for a long time, which is actually difficult to accept, but rather they have a very rapid evolution, and they accumulate mutations much faster than non-miniature fishes. And this is actually cited in the literature for other miniatures. Tyson Roberts, for instance, raised the hypothesis without molecules back at the 70s that these miniature fishes sorry, have a much faster evolutionary rate. And so this would be expected, as you can actually see for their when you look at their genetic differences. So biological diversification in Priorcarricks follows a pattern of mosaic of features, most of them relating to developmental truncation in the skeleton. And so therefore, diagnosis must be done based on the combination of characters, especially these ontogenetic characters. And in order to do that, you need to examine a large amount of specimens of different populations and localities in order to fully understand the patterns of morphological variation within the genus. And of course, you can only access this information if you have good preserved specimens to begin with. So once again, if you catch specimens lying in a jar in the museum collection, as you would do for non-miniature fish, you won't be successful. So the project continues. And I'm just gonna finish highlighting that the Nootropics holds more than 250 miniature species from all major lineages. So we're talking about Classiforms, Siluriforms, Cyprinodontiforms, such as this Phylogenetic, and there's even miniature anchovies here. So they're just waiting for a similar approach to be done. And with that, I have to thank all my collaborators along the last 10 years, because this project was started as a postdoc, and it's been going on for 11 years now. This couldn't be done without the efforts and aid of Ralph Britz and Monica Toledo Pisa, who I'm greatly indebted, and I wouldn't be here if it wasn't for them. And just to catch something from Ralph's talk, he mentions in the beginning that a few people that actually he considers that was a great mentor to him. And I can say without fear that Ralph Britz and Monica Toledo Pisa are my two role models in ichthyology. I also like to thank my funding agencies and the institutions that made all this possible. Thank you.